Think I've been here before.
Now the discussion is about the platyhelminthes and the nematohelminthes. In these non-chordata, the platyhelminthes and nematohelminthes organisms were specially evolved because most of them are parasites. They were evolved from the ctenophores and the cnidarians, but especially those are adapted to live in the host body, and the most of the organs are not developed in these parasites because there is no need to develop the body organs. They are parasites. They can steal the digested food material from the host body because they are living as the endoparasites. Mostly they can live in the small intestine and large intestine where the readily digested food is available. Phylum number 4, the platyhelminthes. Now we start with the general characters of platyhelminthes. Now the first question is habitat. Where do they live? Most of them can live in the aquatic environment. In the aquatic environment, they only the free living platyhelminthes can live in the aquatic environment, but remaining all the trematodes and cystodes, the parasites, can live in the host body. Those are endoparasites. They can live inside the host body. Fasciola hepatica, trematodes and cystodes, tapeworms and other parasitic organisms can live inside the body. Especially those are adapted to live in the small intestine where the readily available digested food, glucose, amino acids, glycerol and fatty acids are available. Body structure. In this body structure, actually I will discuss about the body organization throughout the animal kingdom clearly, but after completing this platyhelminthes and nematohelminthes, while we are discussing about the annelida, I will discuss about the body organization clearly from the protozoa to chordata. I will discuss about the body organization. There is no worry about this. If any point is not understanding, for example, bilateral symmetry, what do you mean by bilateral symmetry? Means there is no need to bother about this because I will discuss about them in the body organization. Here, body structures, just you can try to understand this. The body structure, this is for example free living animal Dugisia and planaria. This is Fasciola hepatica. The body organism dorsal. The main important character of these platyhelminthes are dorso-ventrally flattened body. The body of these platyhelminthes are leaf-like or a paper-like body. There is no body shape. There is no particular body shape, and there is no coelom inside the body. During the embryonic development, the mesoderm occupied the complete space. That's why there is no body coelom inside the body. So the body is like a flattened body, leaf-like or a paper-like body. But they can show bilateral symmetry. Bilateral symmetry means the body can be cut into two halves. That is called a bilateral symmetry. But before this, phylum Cnidaria and sponges and the protozoans, we do not find this type of bilateral symmetry. Radial or biradial or asymmetrical forms we observed in these three Cnidaria, sponges and the protozoans. But now platyhelminthes onwards we can find this bilateral symmetry. And another one is the triploblastic animals. Triploblastic means during the embryonic development, ectoderm, mesoderm and endoderm, 3 layers are present. From these 3 layers, all the organs of the body are developed in the later embryonic development after the gastrulation. That's why these animals are called the triploblastic animals. Platyhelminthes onwards, all the animals are triploblastic animals, including the chordates. We are also triploblastic animals. During the embryonic development, we too have ectoderm, endoderm and mesoderm in our body. Then after the organogenesis take place, due to the organogenesis, all the body organs are formed in later embryonic development. But before these platyhelminthes, Cnidaria and protozoa and porifera, there is no triploblasticity. In Nidaria, those are diploblasticity, because these are only two germ layers are present during their embryonic development, ectoderm and endoderm. There was no mesoderm. That's why a coelenteron is formed in the coelenterate or Nidarians. But in the protozoan sponges, there is no discussion about the ectoderm, endoderm and mesoderm. Those layers are absent during the embryonic development. These are called the acoelomates. What are acoelomates? Because there is no coelom in their body. I said to you already, the body is looking like a flat leaf-like body or paper-like body. Means there is no particular coelom inside the body. There is no body cavity. Why there is no body cavity in them means during the embryonic development, at the time of gastrulation, the mesoderm is developed, but the mesoderm is completely the mesoderm is completely occupying the space in between the ectoderm and endoderm. If the mesoderm is completely occupied the space is not left. There is no space inside the body. There is no cavity inside the body. That's why there is no coelom in the body. At this place, parenchyma is present, the space in between the ectoderm and endoderm. This is the important point. Those are called the acoelomates because there is no coelom. There is no coelom, there is no body cavity. That's why the body is leaf-like, dorso-ventrally flattened body. Organ level of organization. This is another important point in these organisms. Before this, in Nidaria and sponges and protozoans and poriferans, sponges, those are cellular level of organization. The cells are present in the body. The tissue level of organization we do not find in the protozoans and poriferans. But whereas in the Nidarians or coelenterates, we can find some tissues. Tissue level of organization they exhibit. But now, platyhelminthes onwards, organs are present in the body. That is called organ. They can exhibit the organ level of organization. So many organs are present in the reproductive organs and the digestive system, and in some of the free living animals, circulatory system and the ladder like nervous system. Some organs are present in the body. That's why it exhibits the organ level of organization in its body. Size and shape. How is the size? How is the shape? There is no particular size. Dorso-ventrally flattened body, leaf-like. There is no particular shape to them. If we want to discuss about the size, some of them may be microscopic and some are large animals. Some Teniasolium like animals are meters and meters. And the body covering is very important. In free living animals, planaria and Dugisia, for example, this one, planaria and Dugisia, because those are free living animals, they have to move from one place to another place to capture the food material. But in parasites, trematodes and cystodes, the tegument is present, which is made up of a fatty layer called the cuticle. The tegument can protect the parasites from the host enzymatic reaction. For example, if these trematodes and cystodes live in our small intestine, the proteases enzymes may kill them. Proteases enzyme may digest the parasites. If they are digested, those will be died. But if those are died, there is no population of parasites, trematodes and cystodes. That's why, to protect themselves from the enzymatic reactions of host, they are developed a cuticle layer which is made up of fats, that is called the tegument, and protect themselves from the host enzymatic reactions. Digestive system. Is there any digestive system in the platyhelminthes organisms? Yes. In these Dugisia and planaria, those are free living animals. Those are not parasites. They have to capture the food material from the external environment. The food material is in complex form. They have to take them into the body, ingest them into the body. They have to digest with the help of digestive enzymes. So the digestive system is somehow developed, but there is no anus. Only the mouth is located at the anterior region. There is no anus. The digestive system is somehow developed. The food material is ingested into the digestive tract. With the help of enzymes, the digestion is take place. the digested food materials are diffused throughout the body and utilized, assimilated by the body cells. The undigested food material expelled out through the mouth only. This is in the Nidarians also, only mouth is present, there is no anus. And but whereas in the parasites, trematodes and cystodes, there is no particular digestive system in their body. Why there is no digestive system in the trematodes and cystodes? Because those are parasites. Those are parasites, especially those are endoparasites. Endoparasites means most of them can live in the small intestine, few can live in the blood. That's why the blood or the small intestine are providing the readily available food, that is the glucose, amino acids and glycerols and fatty acids. Those food materials are directly absorbed into the body through the body surface directly. Any part of the body can absorb the food material from the surrounding environment, that is already digested food material. That's why there is no particular digestive system in their body. Then what about the circulatory system, for example? There is no particular circulatory system in the in the free living animals. In the parasite, there is no particular digestive system because the food can be transported, food can be diffused throughout the body by the process of diffusion. Excretory system, yes, the excretory system is present in these animals to excrete the excretory product that is called the ammonia. That ammonia is a chief excretory product that can be excreted out. But before that, we had to discuss about the osmotic regulation. This osmotic regulation is nothing but the maintenance of salts and the ionic balance in the body. This especially in this excretory system, these are the flame cells and the solenocytes. Some of the organs, some of the platyhelminthes are containing the flame cells and some are containing the solenocytes. Flame cells, this is a flame like structure, cilia, this is flagella. Yes, the cilia and flagella are also found in these animals. Those are only meant for the process of excretion. We will discuss about the excretion process after a few minutes. Then the excretory system is present in them. This excretory system is especially called as the protonephridia. First time the excretory system is developed in these platyhelminthes. That's why these are calling a first nephridia. Those are excretory organs. Then nervous system. Okay, the development of nervous system is started in these animals. This is a ladder like nervous system is present in the planaria especially, but in the trematodes and cystodes, endoparasites, this nervous system is not highly developed, but a few development we observed in them. But especially in the Dugisia and the planaria, the free living animals needed this ladder like nervous system to respond to the surrounding environment. Then what about this reproductive system? This is very, very important in all the platyhelminthes. The reproductive system is highly developed in the free living animals and as well as in the trematodes and cystodes, in the parasites also. Actually, these are hermaphrodites, bisexual animals. Both the male and female reproductive organs are present in one animal, means one body only. Self-fertilization is taking place. The sperm cells produced by that animal are fusing with the ova produced by the same animal. That is called the self-fertilization. Trematodes and cystodes are fertilizing selfly, but cross-fertilization is common in free-living elements. For example, planaria and Dugisia. This is the cross-fertilization is taking place in those animals. Evolutionarily, cross-fertilization has a lot of significance because in every generation the new type of genetic material is added to the offsprings. But whereas in the parasite, the self-fertilization is taking place. This is very important point. And in the planaria, those are free-living animals, the cross-fertilization is taking place, although they are hermaphrodites. Sexual reproduction and asexual reproduction is also taking place in some of the animals. For example, in planaria, the free-living animal, the fission is the main asexual reproduction, that is actually called as the multiple fission. That is not the binary fission. For example, you can see this is planaria or Dugesia. In these animals, if accidentally any piece of the body is fragmented, every piece can develop one animal. That is called the regeneration. That in the regeneration, every part of the body, every fragment can reproduce the next generations, because every part is containing the totipotent cells and can form the total body. But here the multiple fission means during the asexual reproduction, there are many cells are divided, and some of the cytoplasm is occupied in the cells. The genetic material is separated, and all the cells are separated from the body. These cells can form a spores-like structures, cyst-like or spores-like structures. From the, if the environmental condition is suitable, these spores can develop. Each spore can develop as a full planaria or Dugesia free living animal. That is called the multiple fission. Here the internal fertilization occur in case of sexual reproduction. Parasites form show. Yes, this is very important thing. The complex life cycle. What do you mean by complex life cycle? Why there was a complex life cycle in the platyhelminthes? Because the platyhelminthes organisms, those are either free living animals or the parasites. Those have to face many problems, because if the environmental conditions are not suitable, many of them will die. If they live in the host body, they have to face many harsh conditions, enzymatic reactions, or the immune cells, or temperature. Those many harsh conditions are there, they have to face. So this life cycle is very complex, because the death rate is very high in the platyhelminthes. If the death rate is very high, if they wanted to live, if they wanted to survive, if they wanted to produce the offsprings, the birth rate should be very high. If the death rate is very high, the birth rate also should be very high. That's why they have developed a complex life cycle. For example, in the reproductive system of this, we will discuss about this complex life cycle, how many types of larvae they are producing. From the human body, the eggs are coming out. They can develop as a one type of larva, sporocyst, radia, cercaria, metacercaria, like that. 1, 2, 3 or 4 type of larvae are developing to tolerate the harsh conditions in the external environment or in the host body. That's why they have developed a complex life cycle. Next, our focus is on the excretory organs called the protonephridia. In planaria, these are the flame cells. In other trematodes and cystodes, we find these solenocytes. The difference between the flame cells and solenocytes is flame cells are containing the cilia. This is just like a flame. That's why this is called as these are called as the flame cells. But the solenocytes are containing the flagella. What are the functions of these cilia and flagella present in the flame cells and solenocytes respectively? Because due to the movement of these flame cells, these cilia or these flagella from the surrounding environment. The surrounding environment is actually the parenchyma, the coelom, the parenchyma. From this parenchyma, the excretory material called excess amount of water can enter into this cell due to the movement of the cilia or flagella. Actually, while the cilia or flagella are moving, the negative pressure is created. They can obtain the excess amount of water or excess amount of salts, ions, along with the nitrogenous waste material called the ammonia, are entered into this cell. These are called the flame cells. Our osmotic regulation is achieved by these cells. These are called the special and unique characters of this platyhelminthes. That is very important. Protonephridia or flame cells are solenocytes. These are the excretory organs are present in them. parenchyma, that is, we discussed about this parenchyma, coelom like structure, but this is acoelom, no coelom present in the body. This is completely occupied by the mesoderm during the embryonic development. The trematodes and cystodes, those are endoparasites. They have developed a special organs called the hooks and suckers to attach to the small intestine of host body, because in our body, especially they are living in the small intestine. In the small intestine, there are many problems are there, because while the food is moving, while the water is moving, while the peristaltic movements are taking place, these organisms may be washed out from the small intestine. To avoid these unnecessary movements, they have developed hooks and suckers to attach small intestine firmly, and also tegment. Their body is made up of tough tegment, which is made up of fats, cuticle, to tolerate them from the enzymatic reactions of host. And this is showing hooks and suckers in the trematodes and cystodes. These are called the hooks. With the help of these hooks, they can firmly attach it to the small intestine. These suckers are also helping to attach to the small intestine to prevent the unnecessary movements. Then if we will see the classification of platyhelminthes, class Turbellaria, these are free living animals, Dugisia and Planaria, and the trematodes are flukes. These especially liver fluke called the Fasciola hepatica, for example. And the cystoda, these are called the tapeworms, Taenia solium and Taenia saginata. Some other animals, these are very long animals, sometimes they may be meters and meters. Free living flatworms, those are non-parasitic Turbellarians. Ciliated epidermis, because the cilia are present in their body to move locomotory organs. These are Turbellarians. And regeneration is the very important character in these animals. The Turbellarians is regeneration. The regeneration power is very high. If you cut this animal into many pieces, 5, 6, 10 or 20 pieces, each piece can develop a new animal, because each piece is containing the totipotent cell, or other cells can be converted as the totipotent cell, and the regeneration power is very high. But whereas in the trematodes are endoparasites, the life cycle is very complex. Already we discussed about them, why the life cycle is very complex, because the death rate is very high due to harsh conditions inside the small intestine or outside the body, host body. That's why the complex life cycle is developed them. The tegment is developed in those organisms to prevent them from the enzymatic reactions of host enzymes. Class Cystoda, those are also internal parasite. There is no particular digestive system, circulatory system, respiratory system. No system is present in the parasites, Trematoda and Cystoda, because there is no need of circulatory system, because there is no transportation. There is no need of respiratory system, because they are living in the small intestine. They are living in the blood. There is no oxygen in the blood and in the small intestine. They have to live in the anaerobic condition. They have to perform the anaerobic respiration, and they liberate only very low amount of ATP molecules. With the help of those ATP molecules, they have to run their metabolic activities. That's why there is no particular digestive system, no respiratory system, no circulatory system. Reproductive system is highly developed, and the complex life cycle is developed. That is the life of these platyhelminthes. And another important we have to discuss about this proglatids. Scolex is the anterior part. Proglatids. In these proglatids, there are two types of proglatids we can find in them. In these proglatids, these are called the proglatids. These are segment-like structures are proglatids. At the beginning, this is called the scolex head region. Head region is scolex is containing the hooks and suckers. With the help of those hooks and suckers, they can attach to the small intestine. to avoid the unnecessary movement, but remaining part, neck region or remaining part is containing a immature proglatids. These are matured proglatids. Actually, the sexual reproduction is taking place in these animals, that is self fertilization is taking place. The uterus is filled with the eggs. In the immature proglatids, those eggs are present, but those eggs are not developed. But whereas in this biggest part, the matured proglatids are present, in the developed eggs, matured eggs are present. These matured eggs containing this proglatid is cutting from the body and leaving the organism and entered into the fecal material of that human being or any other sheep or host, that can be excreted out. If those are proglatids are excreted out, if they can reach to the water, they can develop a type of sporocyst like of larva, and they can reach to the snail or other organisms. They can run their life cycle.